Hi friends, my name is Nandita. I am from Andhra Pradesh. Children, today we are going to discuss about mineral nutrition from chapter 12 CBSE syllabus class 11. Children, the basic needs of all living organisms are essentially the same. They require macromolecules such as carbohydrates, proteins and fats, water and minerals for their growth and development. This chapter focuses mainly on inorganic plant nutrition, wherein you will study the methods to identify elements essential to growth and development of plants and the criteria of establishing the essentiality. You will also study the role of essential elements, their major deficiency symptoms, and the mechanism of absorption of these essential elements from this chapter. The chapter also introduces you briefly to significance and the mechanism of biological nitrogen fixation. Children, let us discuss about methods to study the minerals requirement of plants. In 1860, Julius von Schatz, a prominent German botanist, demonstrated for the first time that plant could be grown to maturity in a defined nutrient solution in complete absence of soil. This technique of growing plants in a nutrient solution is known as hydroponics. Since then, a number of improvised methods have been employed to try and determine the mineral nutrients essential for plants. The essence of all these methods involved the culture of plants in a soil-free defined mineral solution. These methods required purified water and mineral nutrient salts. Can you explain why is this so essential? After a series of experiments in which the roots of the plants were immersed in nutrient solution and wherein an element was added, substituted, removed or given in a varied concentration, a mineral solution suitable for the plant growth was obtained. By this method, essential elements were identified and their deficiency symptoms discovered. Hydroponics has been successfully employed as a technique for the commercial production of vegetables such as tomato, seedless cucumber and lettuce. It must be emphasized that the nutrient solution must be adequately aerated to be obtain the optimum growth. What would happen if solutions were poorly aerated? Diagrammatic views of the hydroponic techniques is given in the picture 12.1 and 12.2. Let us study the essential minerals and elements. Children, most of the mineral present in the soil can enter plants through roots. In fact, more than 60 elements of the 150 discovered as so far and found in different plants. Some plant species accumulate selenium and some other gold, while some other growing near nuclear test site take up radioactive strontium. There are techniques that are able to detect the minerals even at a very low concentration. The question is whether all the diverse mineral elements present in a plant, for example gold, selenium as mentioned above, are really necessary for plant. How do we decide what is essential for plant and what is not? Children, let us discuss the criteria for essentiality. The criteria for essentiality of elements are given below. First one is the element must be absolutely necessary for supporting normal growth and reproduction. In the absence of the element, the plant do not complete their life cycle or the set of seeds. Second one is the requirement of the element must be specified and not replaceable by any other element. In other words, deficiency of any one element cannot be met by supplying some other elements. The element must be directly involved in the metabolism of the plant. Hydroponic plant production. Plants are grown in a tube or through placed in a slight insulin. A pump circulates a nutrient solution from a reservoir to the elevated end of the tube. The solution flows down to the tube and returns to the reservoir due to the gravity. Inset shows a plant whose roots are continuously bathed in aerated nutrient solution. The arrows indicates the direction of the flow. Based upon the above criteria, only a few elements have been found to be absolutely essential for growth, plant growth and metabolism. These elements are further divided into the broad categories based on their quantitative requirements. These are called macronutrients and micronutrients. First, let us discuss about macronutrients. Macronutrients are generally present in plant tissues in large amount. The macronutrients include carbon, hydrogen, oxygen, nitrogen, phosphorus, sulfur, potassium, calcium and magnesium. Of this, carbon, hydrogen and oxygen are mainly obtained from CO2 and H2O, while the others are obtained from the soil as mineral nutrition. Let us discuss about micronutrients or trace elements. Micronutrients or trace elements are needed in a very small amounts. These includes iron, magnesium, copper, zinc, boron, chlorine and nickel. In addition to these 17 essential elements named above, there are some beneficial elements such as sodium, silicon, cobalt and selenium. They are required by higher plants. Essential elements can also be grouped into 4 broad categories on the basis of their functions. These categories are essential elements as compounds of biomolecules and hence structural elements of cells. Examples are carbon, hydrogen, oxygen and nitrogen. Essential elements that are compound of energy related chemical compounds in plants like magnesium in chlorophyll and phosphorus in ATP. Third one is essential elements that activate or inhibit enzymes. For example, Mg2 plus is an activator for both ribulose, biphosphate, carboxylase oxygenase and phosphophonyl pyruvate carboxylase, both of which are critical enzymes in photosynthetic carbon fixation. Zn2 plus is an activator for alcohol dehydrogenase and MO of nitrogenase during nitrogen metabolism. Can you name a few more elements that fall in this category? For this, you will need to recollect some of the biochemical pathways you have studied earlier. Some essential elements can alter the osmotic potential of a cell. Potassium plays an important role in the opening and closing of stomata. You may recall the role of minerals as solutes in determining the water potential of a cell. Children, now let us discuss the role of macro and micronutrients. Essential elements perform several functions. They participate in variable metabolic process in the plant cells such as permeability of cell membrane, maintenance of osmotic concentration of cell's cap, electron transport system, buffering action, enzymatic activity and act as major constituents of macromolecules and coenzymes. Various forms and functions of essential nutrients elements are given below. First one is nitrogen. This is the essential nutrient element required by plants in the greater amount. It is absorbed mainly as NO3, though some are also taken up as NO2 or NH. Nitrogen is required by all parts of a plant, particularly the meristematic tissues and the metabolically active cells. Nitrogen is one of the major constituents of proteins, nucleic acid, vitamins and hormones. Next, let us come to discuss about phosphorus. Phosphorus is absorbed by the plants from soil in the form of phosphate ions. Phosphorus is the constituent of cell membrane, certain proteins, all nucleic acids and nucleotides, and it is required for all phosphorylation reactions. Potassium. Potassium is absorbed as potassium ion K+. In plants, it is required in more abundant quantities in the meristematic tissues, buds, leaves, and root tips. Potassium helps to maintain an anion cation balance in cells, and it is involved in protein synthesis, opening and closing of stomata, activation of enzymes, and in the maintenance of the turgidity of cells. Calcium. The plant absorbs calcium from the soil in the form of calcium ions Ca2+. Calcium is required by meristematic and differentiating tissues. During cell division, it is used in the synthesis of cell wall, particularly as calcium pectate in the middle lamella. It is also needed during the formation of mitotic spindle. It accumulates older leaves. It is involved in the normal functioning of cell membrane. It activates certain enzymes and plays an important role in regulating metabolic activities. Magnesium. It is absorbed by the plant in the form of bivalent Mg2+. It activates the enzyme of respiration, photosynthesis, and are involved in the synthesis of DNA and RNA. Magnesium is the constituent of the ring structure of chlorophyll and helps to maintain the ribosome structure. Sulfur. Plants obtain sulfur in the form of sulfate, SO4. Sulfur is present in two amino acids, cysteine and methanoine, and it is the main constituent of several coenzymes, vitamins. Iron. Plants obtain iron in the form of ferric irons, Fe3+. It is required in a large amounts in the comparison to the other micronutrients. It is an important constituent of proteins involved in the transfer of electrons like ferridoxin, cytochrome. It is reversibly oxidized from Fe2+ to Fe3+ during the electron transfer. It activates catalase enzyme and it is essential for the formation of chlorophyll 2. It is absorbed in the form of manganese irons Mn2+. It activates many enzymes involved in the photosynthesis, respiration, and nitrogen metabolism. The best defined function of manganese is in the splitting of water to liberate oxygen during photosynthesis. Zinc. Plants obtain zinc as Zn2+ ions. It activates various enzymes, especially carboxylases. It also needed in the synthesis of oxygen. Copper. Next, copper. It is absorbed cupric ions. It is essential for all the metabolism in plants. Like iron, it is associated with certain enzymes involved in redox reactions, and it is reversibly oxidized from Cu+ to Cu2+, etc. Boron. It is absorbed in BO33. Boron is required for uptake and utilization of Ca2+, membrane functioning, pollen germination, cell elongation, cell differentiation, and carbohydrate translocation. Molybdenum. Plants obtained it in the form of molybdate ions. It is a component of several enzymes including nitrogenous and metabolism. Last one is chlorine. Chlorine is absorbed in the form of chloride anion Cl along with Na+ and K+. It helps in determining the solute concentration and the ionization balance in the cells. It is essential for the water splitting reaction in photosynthesis, a reaction that leads to oxygen evolution. Children, now let us discuss about deficiency symptoms of essential elements. Whenever the supply of an essential elements become limited, plant growth is retarded. The concentration of the essential elements below which plant growth is retarded is termed as critical concentration. The element is said to be deficient when present below the critical concentration, since each element has one or more specific structural or functional role in plants in the absence of any particular element. Plants show certain morphological changes. These morphological changes are indicative of certain element deficiencies and are called deficiency symptoms. The deficiency symptoms vary from element to element and they disappear when the deficient mineral nutrient is provided to the plant. However, if deprivation continues, it may eventually lead to the death of the plant. The part of the plant that shows the deficiency symptoms tend to appear first in the older tissue. For example, the deficiency symptoms of nitrogen, potassium, and magnesium are visible first in the scent leaves. In the older leaves, biomolecules containing these elements are broken down, making these elements available for mobilizing to younger leaves. The deficiency symptoms tend to appear first in the younger tissues whenever the elements are relatively immobile and are not transported out of the mature organs. For example, elements like sulfur and calcium are the part of a structural component of the cell, hence are not easily released. This aspect of mineral nutrients of plants is of a great significance and importance to agriculture and horticulture. The kind of deficiency symptoms shown in plants include chlorolysis, necrosis, stunted plant growth, premature fall of leaves and buds, and inhibition of cell division. Chlorolysis is the loss of chlorophyll leading to yellowing in leaves. This symptom is caused by deficiency of elements like N, K, Mg, S, Fe, Mn, Zn, and Mo. Likewise, neurosis or death of tissues, particularly leaf tissue, is due to the deficiency of Ca, Mg, Cu, K. Lack or low level of N, K, S, Mo causes an inhibition of cell division. Some elements like NSMO delay flowering in their concentration in plants is low. You can see from above that the deficiency of any element can cause multiple symptoms and that the same symptoms may be caused by the deficiency of one of several different elements. Hence, to identify the deficient element, one has to study all the symptoms developed in all the various parts of the plant and compare them with the available standard tables. We must also be aware that different plants also respond differently to the deficiency to the same element. Children, now let us discuss about toxicity of micronutrients. The requirement of micronutrients is always in low amount, while their moderate decrease causes the deficiency symptoms and the moderate increase causes toxicity. In other words, there is a narrow range of concentration at which the elements are optimum. Any mineral ion concentration in tissues that reduces the dry weight of tissue by about 10% is considered toxic. Such critical concentrations vary widely among different micronutrients. The toxicity symptoms are difficult to identify. The toxicity levels for any element also vary for different places. Many a time excess of an element may inhibit the uptake of another element. For example, the prominent symptom of manganese toxicity is the appearance of brown spots surrounding by chlorotic veins. It is important to know that manganese competes with iron and magnesium for uptake and with magnesium for binding with enzymes. Manganese also inhibits calcium translocation in shoot apex. Therefore, excess of manganese may in fact induce deficiencies of iron, magnesium, and calcium. Thus, what appears as symptoms of manganese toxicity may actually be the deficiency symptoms of iron, magnesium, and calcium. Can this knowledge be of some importance to be a farmer, a gardener, or even for you in your kitchen garden? Now let us discuss about mechanism of absorption of elements. Children, much of the studies on mechanism of absorption of elements by plants has been carried out in isolated cells, tissues, or organs. These studies revealed that the process of absorption can be demarcated into two main phases. In the first phase, an initial rapid uptake of ions into free phase or outer phase of cells. The apoplast is passive. In the second phase of uptake, the ions are taken in slowly into the inner phase, the symplast of the cells. The passive movement of ions into apoplast usually occurs through ion channels, the transmembrane protein that functions as selective pores. On the other hand, the entry or exit of ions to and from the symplast requires the expenditure of metabolic energy, which is an active process. The movement of ions is usually called flux. The inward movement into the cell is influx and the outward movement efflux. You have read the aspects of mineral, nutrients take up and translocation in plants in chapter 11. Children, now let us discuss about translocation of solutes. Mineral salts are translocated through xylem along with the ascending streams of water, which is pulled up through the plant by transpirational pull. Analysis of xylem sap shows the presence of mineral salts in it. Uses of radioisotopes of mineral elements also substantiate the view that they are transported through the xylem. You have already discussed the movement of water in xylem in chapter 11. Children, now let us discuss about soil as reservoir of essential elements. Majority of the nutrients that are essential for the growth and development of plants become available to the roots due to the weathering and broken down of rocks. This process enriches the soil with dissolved ions and inorganic salts. Since they are derived from the rock minerals, their role in plant nutrition is referred to as mineral nutrition. Soil consists of a wide variety of substances. Soil not only supplies minerals but also absorbs nitrogen fixing bacteria, other microbes, holds water, supplies air to the roots and act as a matrix that stabilizes the plant. Since the deficiency of essential minerals affect the crop field, there is often a need for supply them through fertilizers. Both macronutrients NPK, S, etc. and micronutrients Cu, Zn, Fe, Mn, etc. form components of fertilizers and are applied as per need. Children, now let us discuss about metabolism of nitrogen. Let us see through nitrogen cycle. Apart from carbon, hydrogen and oxygen, nitrogen is the most prevalent element in living organisms. Nitrogen is a constituent of amino acids, protein, hormones, chlorophyll and many of the vitamins. Plant complete with microbes for the limit nitrogen that is available in soil. Thus, nitrogen is a limiting nutrient for both natural and agricultural ecosystem. Nitrogen exists as two nitrogen atoms joined by a very strong triple covalent bond N is equal to N. The process of conversion of nitrogen N2 to ammonia is termed as nitrogen fixation. In nature, lightning and ultraviolet radiation provide enough energy to convert nitrogen to nitrogen oxides NO, NO2, N2O, etc. Industrial combustions, forest fires, automobile exhausts and power generating stations are also sources of atmospheric nitrogen oxides. Decomposition of organic nitrogen of dead plants and animals into ammonia is called ammonification. Some of this ammonia volatilizes and re-enters the atmosphere, but most of it is converted into nitrate by soil bacteria in the following steps. Ammonia is the first oxidized to nitrate by the bacteria Nitrosomonas and Nitrococcus. The nitrate is further oxidized to nitrate with the help of bacterium Nitrobacter. These steps are called nitrification. These nitrifying bacteria are chemoautotrophs. The nitrate thus formed is absorbed by the plants and it is transported to the leaves. In leaves it is reduced to form ammonia that finally forms the amine group of amino acids. Nitrate present in the soil is also reduced to nitrogen by the process of denitrification. Denitrification is carried by a bacteria, Pseudomonas and Thiobacillus. Children, let us discuss about biological nitrogen fixation. A very few living organisms can utilize the nitrogen in the form of N2 available abundantly in the air. Only certain prokaryotic species are capable of fixing nitrogen. Reduction of nitrogen to ammonia by living organisms is called biological nitrogen fixation. The enzyme nitrogenase, which is capable of nitrogen reduction, is present exclusively in prokaryotes such as microbes, are called NH2 fixers. The nitrogen fixing microbes could be free living or symbiotic. For examples of free living nitrogen fixing aerobic microbes are Azotobacteria and Bigernica, while Rhodospirillum and anaerobic and free living. In addition, a number of cyanobacteria such as Anabinia and Nostoc are also free living nitrogen fixers. Symbiotic biological nitrogen fixation. Several types of symbiotic biological nitrogen fixing associations are known. The most prominent among them is the legume bacterial relationship. Species of rod-like rhizobium has such a relationship with the roots of several legumes such as alfalfa, sweet clover, sweet pea, lentils, garden pea, broad bean, clover beans, etc. The most common association on root is as nodules. The nodules are small growth on the roots. The microbe Frankia also produces nitrogen fixing nodules on the roots of non-leguminous plants. Both rhizobium and Frankia are free living in the soil but as symbionts can fix atmospheric nitrogen. Uproot any one of plant a common pulse just before flowering. You will see near spherical outgrowths on the roots. These are nodules. If you cut through them, you will notice that the central portion is red or pink. That makes the nodules pink. This is due to the presence of leguminous hemoglobin or leg hemoglobin. Children, let us discuss about nodule formation. Nodule formation involves a sequence of multiple interactions between rhizobium and root of the host plant. Principal stages in the nodule formations are summarized as follows. Rhizobia multiply and colonize the surroundings of roots and get attached to the epidermal and root hair cells. The root hair curl and the bacteria invade the root hair. An infection thread is produced carrying the bacteria into the cortex of the root, where they initiate the nodule formation in the cortex of the root. Then the bacteria are released from the thread into the cells, which leads to the differentiation of specialized nitrogen fixing cells. The nodule, those formed, establishes a direct vascular connection with the host of exchange of nutrients. These events are depicted in figure 12.4. The nodule contains all the necessary biochemical compounds such as the enzyme nitrogenase and leg hemoglobin, etc. The enzyme nitrogenase is a MOFE protein and catalyzes the conversion of atmospheric nitrogen to ammonia, the first stable product of nitrogen fixation. Children, just look at the examples of development of nodules in a soya bean. In picture A, rhizobium bacteria contact a susceptible root hair, divide near it. In picture B, successful infection of the root hair causes it to curl. In infected thread carries the bacteria to the inner cortex. The bacteria get modified into rod-shaped bacteriodes and cause inner cortical and pericycle cells to divide. Division and growth of cortical and pericycle cells lead to nodule formation. In deep image D, you can see a mature nodule is complete with a vascular tissues continuous with those of the root. Children, the enzyme nitrogenase is highly sensitive to the molecular oxygen. It requires anaerobic conditions. The nodules have adaptations that ensure that the enzyme is protected from oxygen. To protect these enzymes, the nodule contains an oxygen scavenger called leg hemoglobin. It is interesting to note that these microbes live aerobes under free living conditions where nitrogenous is not operational, but during nitrogen fixing events they become anaerobic. You must have noticed in the above equation that the ammonia synthesized by nitrogenous requires a very high input of energy. The energy required those is obtained from the respiration of the host cells. Fate of ammonia 80 physiological pH. The ammonia is protonated to form NH plus 4 ion. While most of the plants can assimilate nitrate as well as ammonium ions, the latter is quite toxic to plants and hence cannot accumulate in them. Let us now see how NH4 plus is used to synthesize amino acids in plants. There are two main ways in which this can take place. One is reductive amination. In this process, ammonia reacts with beta ketogluteric acid and forms glutamic acid as indicated in the equation. Second one is transamination. It involves the transfer of amino group of one amino acid to the keto group of keto acid. Glutamic acid is the main amino acid from which the transfer of NH2, the main group, takes place and the other amino acids are formed through transamination. The enzyme transaminase catalyzes all such reactions. For example, the two most important amides, asparagine and glutamine, found in plants are a structural part of proteins. They are formed from two amino acids, namely aspartic acid and glutamic acid respectively, by addition of another amino acid group to each. The hydroxyl part of the acid is replaced by the another NH2 radical. Since amides contain more nitrogen than the amino acids, they are transported to other parts of the plant via xylem vessels. In addition, along with the transpiration stream, the nodules of some plants export the fixed nitrogen as urites. These compounds also have a particularly high nitrogen to carbon ratio. Children, let us summarize the lesson. Children, plants obtain their inorganic nutrients from air, water and soil. Plants absorb a wide variety of minerals. Not all the minerals elements that they absorb are required by plants. Out of the more than 105 elements discovered as so far, less than 21 are essential and beneficial for normal plant growth and development. The elements required in a large quantities are called elements, while those required in less quantity are called as micronutrients. These elements are either essential constituents of proteins, carbohydrates, fats, nucleic acids, etc. Children, the deficiency these essential elements may lead to symptoms called deficiency symptoms. Chlorosis, necrosis, stunted growth, impaired cell division, etc. are some prominent deficiency symptoms. Plants absorb minerals through the roots by either passive or active process. They are carried to all parts of the organism through xylem along with water transport. Nitrogen is very essential for the sustenance of life. The plant cannot use atmospheric nitrogen directly, but some of the plants in association with N2 fixing bacteria, especially roots of legumes, can fix this atmospheric nitrogen into biological usable forms. Nitrogen fixation requires a strong reducing agent and energy in the form of ATP. N2 fixation is accomplished with the help of nitrogen fixing microbes, mainly rhizobium. The enzymes nitrogenase, which plays an important role in biological N2 fixation, is very sensitive to oxygen. Most of the process takes place in anaerobic environment. The energy ATP required is provided by the respiration of the host cells. The ammonia produced following into fixation is incorporated into amino acids as the amino groups. Thank you.